Nuclear pores are large protein complexes that cross the nuclear envelope, which is the double membrane surrounding the eukaryotic cell nucleus. There are about an average of 2,000 nuclear pore complexes in the nuclear envelope of a vertebrate cell, but it varies depending on cell type and the stage in the life cycle. The proteins that make up the nuclear pore complex are known as nucleoporins. About half of the nucleoporins typically contain solenoid protein dormants, either an alpha solenoid or a beta propeller fold, or in some cases both as separate structural domains. Each NPC contains at least 456 individual protein molecules and is composed of 30 distinct proteins. The other half show structural characteristics typical of natively unfolded, or intrinsically disordered proteins, that is, they are highly flexible proteins that lack ordered secondary structure. These disordered proteins are the FG nucleoporins, so called because their amino acid sequence contains many phenylalanine glycine repeats. Nuclear pore complexes allow the transport of the molecules across the nuclear envelope. This transport includes RNA and ribosomal proteins moving from nucleus to the cytoplasm and proteins, carbohydrates, signaling molecules and lipids moving into the nucleus. It is notable that the nuclear pore complex can actively conduct 1,000 translocations per complex per second. Although smaller molecules simply diffuse through the pores, larger molecules may be recognized by specific signal sequences and then be diffused with the help of nucleoporins into or out of the nucleus. Interestingly, it has been recently shown that these nucleoporins have specific evolutionary conserved features encoded in their sequences that provide insight into how they regulate the transport of the molecules through the nuclear pore. Nucleoporin-mediated transport is not directly energy requiring, but depends on concentrations gradients associated with the RAN cycle. Each of the 8 protein subunits surrounding the actual pore projects a spoke-shaped protein over the pore channel. The center of the pore often appears to contain a plug-like structure. It is yet unknown whether this corresponds to an actual plug or is merely cargo caught in transit. Size and complexity. The entire nuclear pore complex has a diameter of about 120 nanometers in vertebrates. The diameter of the channel ranges from 5.2 nanometers in humans to 10.7 nanometers in the frogs and Opus liveis, with a depth of roughly 45 nanometers. mRNA, which is single-stranded, has a thickness of about 0.5 to 1 nanometer. The molecular mass of the mammalian NPC is about 124 megadaltons and it contains approximately 30 different protein components, each in multiple copies. In contrast, the yeast Saccharomyces cerevisiae is smaller, weighing only 66 mDa. Transport through the nuclear pore complex. Small particles,